us this okay.
Thank you very much. Well, thank you very much for this introduction and to give me the opportunity to present here my work. I wanted to start my talk just as giving a little of background of what I was doing it. As Godot maybe mentioned, I did my career in the University of Valencia. I am a biologist. After my career, I did the master with José Antonio Godoy. And after that, I think that I consider that it was my golden year because I got collaboration with so many projects here in Doñana. So I got collaboration with Carlos Camacho and Basti in a nice project with Niger. After that, I also got collaboration with Ivan Gomez and Rosa Rivas. We were working in Doñana during a few months with tadpoles. Apparently, Carlos Camacho liked how I work, and I went to work with him again and with Jaime Pote in Madrid with fly catchers. After that, I went to France, and I was working with dolphins in a project that was super nice. And in the midtimes, I come back to Seville, as Jose mentioned, to present my work in the conference, where I had the luck to meet my wonderful supervisor, Luis Arseni, that gave me the opportunity to do the PhD with her in Birmingham, and I was studying there climate change on natural populations. So I'm going to tell you a little more about that. And let's introduce my project a little. As you can see, in the last 200 years, the human population has increased from 1 billion to 8 billion of people. And that just means that we need more of everything. We need more food, more space, more natural resources. And if we need more food and more space, what we do is decrease our forests. So since 2000, the 27% of our forests have decreased. And we also increase a lot of the chemical that we use in our land because we need to grow our food. So we use a lot of chemistry in terms of pesticides, herbicides, fertilizers. And that has a really bad effect in lakes because it can be contaminated with these chemicals. And just in the last 50 years, the 50% of our lakes have been contaminated with eutrophication. And I don't know if you realize, but basically every single human in the planet depends on lakes. So just to refresh a little the eutrophication concept, what does this mean is that the lakes get enrichment with nutrients, mainly with nitrogen and phosphorus. And that makes really happy the algae that have limited resources to grow. So they grow massively and cover the surface of the lake. But during the winter, this mass of algae sink to the bottom of the lakes. And they are decomposed by the bacterium community, where it can consume the entire oxygen of the lake. And if we don't have oxygen, there is not a possibility for other species to live on it. And on the other side, we realize also that the temperatures in the planet have increased. I think that Seville is really warm in the last years. And you can see here how the global temperature increased, but not just that. The temperature anomalies in terms of heat waves have also increasing. And you can see in this small video that the blue lines have been replaced for red lines that are the heat waves in the planet until basically get a full red circle. So the question is how those changes can affect the species or how they can face these dramatic changes. They had three options. The first one is that they don't do it, so they got extinct. In this plot is a plot from the WWF report, and they basically study almost 4,000 species, and from all of them, more than the half had decreased in the last years. They can also, if they are the possibility, they can also move or polar shift. And here, for example, Camille Parmesan published a really nice paper where you can see that one population of butterfly moved from the south of UK until the north of UK with the times, and that is correlated with increase of temperatures. And finally, what we can do if they cannot move, what they can do is adapt or change use plasticity to adapt to these changes. I'm not gonna explain what is evolution because everybody knows, but I'm gonna remember a little what is phenotypic plasticity, and it's just a change in behavior, morphology, or physiology in the same organisms and in the same generation. So here I put a nice example of 4 different species of Daphnia. In the left side, you can see one individual that had not been exposed to predators, and in the right side, you see the same individual but exposed to predators, and you can see how in the same generation they can modify massively the morphology. So the problem is that study evolution is really tricky. So you can follow a species during so many times that it becomes really difficult because sometimes follow a natural population during many years is not possible. You can use a species with some generation time, as for example here that you can use bacterium, and you can expose them to a common garden experiment, exposing them to different temperatures. You can use also space for time substitution that basically is collect the same species in 2 different regions, and after again do common garden experiments and compare the reaction norms of all of them in the original habitat or in the different one. But all of them have limitations. So it's not always possible to follow a species during so many years. So generation times you are limited because you cannot study natural population, or space for time substitution, it can be really biased. So coming back to the title of my presentation, how we can study 100 years of evolution in one afternoon, and the response is resurrection ecology. So basically, the resurrection ecology is the study of evolution and climatic change across many generations. It has 2 main components. The first one is sediment cores, and the second one is diapason eggs. I'm going to start explaining a little about sediment cores. So the sediment cores are just natural archives of population stories, and the new technologies and the advance in the analysis of sediment cores have been really, really good in the last year because we can extrapolate from the sediment cores so many information in terms of temperature, concentration of carbon, oxygen, even humidity. And also, they are important that we can date the sediment and know exactly which year it belongs each layer, and they contain X banks, or they are considered as X banks that I will explain in a little bit. So what we do, or what I did during my thesis, is go to the middle of one lake where we have a lot of information about the environmental change. That is a team that is specialized in doing that. They go to the middle of the lake, they collect the sediment core, they bring back the sediment to the land. And after, what we do is place the sediment in this little structure, and we are going to collect one layer that it belongs to 1 centimeter per time. So basically, with this little part in the core, they push up the sediment, and every time that they push up, we collect one layer of sediment. And after, you can store the sediment at 4 degrees and have it there for as long as you want. So we have our sediments, we collect our bag, and after we date the sediments, we analyze the sediment to collect environmental traits, and we also recover all the diapause eggs that are also called in amphibia, and that I'm going to explain now. So basically, if you are really interested in how it has been performed, I think we published this protocol in JOVE, that is the Journal of Visualized Experiments, and you can see the video as well as see the protocol. So just to continue, I wanted to explain a little now how it's possible, which is the species that produce diapause eggs. So basically, most of the Daphnia species in lakes are producing diapause eggs or resting eggs. They are really, really important for lakes. There are key species in lake. We have also genomic resources, because I think that John Colbert published in 2011 the first genome of Daphnia pulex. Now we have also Daphnia pulex, magna, and galeata. They are really high sensitive to environmental change, and they are really easy to grow in the lab, and they have a short generation time. So what we do is, once that we have our sediment, we collect our egg, and yes, we decapsulate the eggs, as I'm showing here. So it's quite tricky because you can pop the eggs really easily. So with a lot of care, you try to put out the 2 eggs that you have in each ephepium. And after, what we do is collect the eggs, and we put them in a normal condition. That is basically light and food. And when they are in normal conditions, also with temperature, they start to grow, to move every time, to move a little faster and trying to expand the egg. And after, finally, they hatch. Pop. So after I start to move as crazy, and I slow down a little the video because it was almost impossible to catch it. Okay, so and finally, the most important characteristic of Daphnia is that you have the ability, they have clonal reproduction and sexual reproduction. And it's great because with clonal reproduction, you have illimited material from the same individual, and you can expose them to different environments, and at the same time, you can quantify the plasticity of these individuals. And at the same time, with a dormant eggs or ephepium, we can compare past and recent population, and we can study the evolutionary mechanisms of these populations. And on the other side, once that you have the ideal model organisms, where you have the ideal also system, and in my case it was Lake Rank case in Denmark, we have a lot of information about this lake because there is a group in Belgium, Luc Dimester, that is working with this lake, and they collect information about that, and also we can collect information from the sediment. So what do we know? We know that the temperature has increased 1 degree, roughly 1 degree in the last 100 years. It was a pristine lake until the 50s. After, in the 50s, the closest village that you can see in the right of the picture, produced a massive eutrophication because they decided to get rid of the dirty water on the lake. After that, the seawater inflow, it was diverted, but we have a massive contamination of pesticides. One of the most common pesticides in Denmark, it was carbaryl, that it was banned after the 80s because it was cancerogenous for humans. And after, in the 2010, we can consider that the lake has been recovered, but the temperature and the frequencies of heat waves has increased. So what we did is recover our 3 subpopulations from our sediment. And after, what we want to know is how this dramatic change has affected the evolution of our population. So the question of my thesis, it was, after all of these changes, are the species able to survive to the environmental change? If they do it, if they are able to survive, how they do it? And if we combine different stressors in this natural population, are they able to survive in that case, or how they are going to perform? And to answer these 3 questions, I used Daphnia magna as model species. So, yes, briefly, I'm gonna explain what I did in my experiment. So, again, I have 3 populations, and I exposed them to 2 temperatures. So, 18 degrees is gonna be. 18 degrees is the temperature of the lake, so it's not as stressful for them. But 24 is the stressful temperature for the individuals, and it's the temperature predicted by the Intergovernmental Panel of Climate Change for the next 100 years. And after, what we did is combine these 2 temperatures with different stressors. That in my case, it was food and pesticides because it's the stressor that they were in the lake at the time. And after all these experiments, I collect data from life history traits, so the typical traits like size, fecundity, mortality, CT-max, that I will explain a little. And age of maturity, I collect also molecular traits like histoproteins or hemoglobin concentration, and also data from genomic and transcriptomic. And briefly, I'm going to mention what's a reaction genome. I think that most of you are familiar with that. But the reaction genome is just to see how the animal performs in terms of plasticity or evolution in our 3 populations. So you can see in the first case that they perform the same, but they evolve in time. In the second time, you can see that there is not plasticity. And in the third time, you can see that there is one interaction between the environment and the genotype. And we analyze the data with simple linear model, we are going to have three components in our models. So the first one is going to represent the evolution, and the temperature and the food or insecticide is going to represent the plasticity in our models or in our population. And finally, I also use phenotypic trajectories analysis that I don't know if you're familiar with, but it's a really nice technique because you can analyze all the phenotypic traits or molecular traits that you have at the same time. So it's running one MANOVA, basically. So it had two main components, the magnitude, that is the difference in length between two populations, and also the direction, that is the direction between the lines. That is going to represent our temperature, but I will explain a little more with a result. And as results, so the first question in my project is if the population are able to survive to those chains. Again, we have our three populations, and here I represent the mortality. By color are going to be represented the three populations, the full lines at 80 degrees, and the dashed line at 24 degrees. And after you have the 100% of mortality, and in the x-axis you are going to have the days of the days of the experiment. So I have my temperature combined with food limitation, or temperature combined with insecticides, and you can see that in the first plot there is just temperature. The animal can do it, so they survive to these chains. If you expose them to temperature combined with food limitation, fine, they don't care, they survive. However, when you expose them to insecticide, I have a 100% of mortality in one of my populations that it was also the population that in the past suffered pesticides. And when you expose them to high temperature, they seem to perform better, but that has an explication, and it's because the carbaryl, that is the insecticide that I use, degrades with high temperature. So at 24 degrees, they have less carbaryl than in fact at 18 degrees. So as conclusion of this part is Daphnia population can survive, but this survival depend of the stressor. And it's normal that when you expose to food, they more or less can do it because they are used to have more or less food. But when you expose to one abiotic stressor, as it can be a pesticide that is really targeting, they cannot cope and they die. So the second question is, which are the evolutionary mechanisms that they use? Again, my three populations. And here what I want to show is some of results of Ctmax. So Ctmax is the maximum tolerance of temperature that this population can reach. So what we did basically is, after all my experiment, we exposed them, we increased the temperature of the water until they stop to move, and then we record the temperature. And we have in the y-axis the temperature, and the x-axis we have our temperature experiment, 18 and 24. And here I represent again the temperature and food limitation, and temperature of insecticides. So if we focus just on temperature, we can see that the most recent population, that is the red line, is the one that performed better. And besides, those changes are significant, so mainly that the population term in my model were significant, as well as the temperature. When we combine with food limitations, these population terms, it doesn't come significant anymore, but temperature is still significant. And if we combine temperature and insecticide, the population is not significant neither, but just the temperature. So at the conclusion of the part, we can say that when we expose just to temperature, they need to have a genetic and a plastic compounds to respond on it. But when we combine different experiments, they use just plasticity. So in fact, they use a combination between genetic and plastic factor to respond to Ctmax. And finally, my third question was, okay, are the response of our population modified if we combine different stressors? And here I represent the fecundity of my population. So again, we have our three populations, the fecundity in the y-axis, and the x-axis we have our temperature, and we have again temperature and temperature combined. In that case, I represent the high concentration of food, and the low concentration of food. High concentration is the dust line, and the low concentration of food is the food lines. And the same for insecticide. The dust line represents the high concentration of pesticides, and the food lines represent the low concentration of pesticides. And we can see that for the temperature, more or less they perform. However, at 24 degrees, you can see that my 3 populations have less neonates. So it's fact that, in fact, they can manage. So they are happy. They are quite happy in temperature. But if you increase too much the temperature, you are going to have an effect on fitness in these individuals. When we combine with food, you can see that temperature is not an effect anymore. So when they have a lot of food, they are happy, they don't care about temperature. And when they are really limited in food, they are really unhappy, but they don't care about temperature neither. And in combination with insecticides, for the most recent population, the 3 population responds better at 24 degrees because, as I mentioned before, at 24 degrees, the carbide degrades. So, as conclusion in this part, what I want to say is that you cannot, or in general, the studies cannot extrapolate the consequences of climate change just using temperature as the main stressor, because in fact, in nature, you never have just temperature. You have a combination of stressors, and if you really want to know what is happening, you need to perform an experiment that in fact combines several stressors. And as a final result, I wanted to show just hits of protein, because I think that this analysis is really nice. So we use 4 different kinds of histoproteins to do our analysis. And here, as I mentioned before, you are going to have 2 components, the magnitude, that is the length of the lines, and the direction of the lines. So here, for example, you see that the 3 lines are the same in length, but 2 populations perform completely different direction than the blue line. So that's why you have that the direction is significant between populations. The full dots are going to represent our control, and the empty dots represent our treatments. And as you can see here, if we expose them to 18 degrees, that is the normal temperature that they have, just exposing to 18 degrees, they are happy, there are no difference between populations. However, when we combine temperature with another stressor, that in that case it was food limitation, only the population that have live in eutrophication in a eutrophication environment, that is the blue line, is the one that performed different. And at the same time, when you combine 80 degrees with pesticides, you can see that the blue line, that is the one that have not experienced this kind of pesticides, is the one that perform better. However, what happens when we combine with a second stressor? So what it happened is that if it's just temperature, the blue line, again, that is the oldest population and the one that doesn't experience heat waves, is the one that performs different. But when you have combination between 24 and food limitation or insecticide, it becomes a little more messy. So again, you cannot predict what can happen when you combine a stressor from a single stress. And you can see at phenotypic level, but also a molecular level, that is the case. Ongoing work. So I finished my thesis, but not my work, because science never had end. And what I did is during my thesis, so during my experiment, I collect the phenotypic trace in a single jar, but at the same time, I collect 400 transcriptome across all my experiments. So that it was a massive work, because I have three incubators of this size several times. But it's nice because at the end we have a lot of data. So what we have is that we had the genome of all the individuals of my population, and we have also the environmental traits that we have a lot in terms of temperature, insecticides in the environment, and so on. And also what we have is the phenotypic traits in terms of life history traits, transcriptome, metabolomes, epigenomes. So normally what we can do is perform a GWAS where you can correlate genotype with environment or genotype with phenotype. However, what we are trying to do is combine the 3 of them. So we want to to do an analysis where it tell us, okay, tell me the most important trait from the environment, the most important trait from the genome, and the most important trait of my life history traits, or phenotypic traits, combine them, and after tell me what is happening. And that is developing in research in eLab. And basically, there are my data, but I don't have the time to analyze all of them, so we are in collaboration with the bioinformatics team there. And I just wanted to finish giving advice to the PhD students if they are here. So I think that if you are looking for a PhD position, my advice is like, don't take the first project that you find it just because it has been just given to you. Be clear that you want to do this project because you are going to spend 4 years maybe analyzing the same data, and you need to be happy doing that because it's 4 years of your life and so much time. If you are starting your PhD project, the other day I was reading a nice opinion article from Susan Vogel in Nature, and it's the 20 things I wish I will know when I started my PhD. It's really simple, but I think that you should read it because it comes with points like organize yourself, read a lot of bibliography. It's never too early to start writing your thesis, and that's completely true. And finally, if you are finishing your PhD and looking for your next position, my advice is start to look for postdoc before you finish your thesis. Because if you don't do it, you are going to find yourself in the situation that you, after being outside of your house so many years, you come back to your house, your parent house with 30 years old, you are looking for position, you don't find anything, you get desperate, and you say, okay, I give up science. But that is something that you shouldn't do because I think that science it can be really, really nice. And if you do a PhD, it's because you know how to do it. So don't give up and just continue trying. And thank you very much.